Conjugation is a mechanism of gene transfer that requires direct contact between donor and recipient cells. A plasmid is a small piece of DNA, separate from the main chromosome, that carries genetic information for such things as antibiotic resistance. The first step in plasmid transfer is contact between the donor and the recipient. The pilus of the donor cell recognizes and binds to specific receptor sites on the cell wall of the recipient cell. The plasmid then becomes mobilized for transfer when an enzyme cleaves one strand of the plasmid at a specific nucleotide sequence, called the origin of transfer. A single strand of the plasmid, beginning at the origin of transfer, enters the recipient cell. Once inside the recipient cell, a complementary strand to the single DNA strand is synthesized. When donor and recipient cells are mixed together, eventually all of the cells become donors.
So I call these my slutty bacteria because they, when one of them has what they call a fertility or resistant plasmid, they will make all of the cells that are not, virginal cells, if you want to call them that. All of them that don't have, they're like F- cells or R- cells, they'll make every one of them resistant or have that copy of that plasmid. So one bad apple spoils the whole bunch. That's the deal. And that's how we get antibiotic resistance in many cases. So it's really important to take your antibiotics as directed. So the S layer are thousands of copies of a single protein linked together, and they are providing protection from environmental conditions. So when it's a hostile environment, the S layer will be produced and it helps the bacteria survive. So what I understand is that typically these are produced in bacteria that do not produce an endospore. So an endospore is also made for survival, and they're only going to be made when it's in a hostile environment. So those cells that do not have the ability to make an endospore use the S layer as this extra layer of protection. The glycocalyx is then deep to that. It is the next layer down, and it's repeating subunits of polysaccharides, not proteins. And they may or may not include protein, but it's repeating polysaccharide typically. The slime layer is glycocalyx that's loosely formed around the cell, and it protects from loss of water and nutrients. The capsule is more tightly bound than the slime layer. It's denser, more organized, and thicker than the slime layer. So you can see here the slime layer kind of loosely organized around, and then here's the capsule that is more thick and dense. And we'll be doing a capsule stain in lab. So you're going to be able to see the cell stained and the background will be stained, and the capsule, for whatever reason, does not take up the dye. It must be pretty neutral, and so it doesn't take up the dye. It's not charged at all. So you can see a picture of the encapsulated bacteria here. The capsule is clear. The bacteria cell is kind of a pink color, and the background is sort of blue. This is similar to what you're going to see. It depends on the bacteria that you use with the capsule stain, but this is a typical example. And then when you look at it on the plate, you can know that it's a encapsulated bacteria because it has kind of a mucoid or snot-like appearance. So you can see it's kind of slimy looking, kind of like snot on the plate. Where these have real distinct, easily seen individual cells, these are all kind of fused together even on the plate here. So capsules are going to be formed by a lot of pathogenic bacteria, and they're important to us because of that. They are able to protect the bacteria from phagocytic white blood cells, and they form these colonial biofilms as well. So like plaque on teeth protect the bacteria and kind of form this community that supports each other. So one bacteria may be able to break down a layer of the nutrients, and then the other layers can use the products of what they broke down as food. And they may have different areas that are anaerobic and other areas that are aerobic. And so we'll look at an animation of this in just a minute. But they also are found on indwelling artificial devices. So this is a real concern if you get a knee, hip, shoulder replacement with those artificial devices. They can form this biofilm, and it's about 1,000 times more resistant to chemicals, antibiotics, and antiseptics and other things than other cells, other bacteria cells. So it is very difficult to kill, and a lot of times with those knee replacements, they have to go back in and replace the knee, have to dig out and debride it if it's infected, and then they have to replace the artificial device, which is why they have to do such a big cleansing ritual before the actual surgery. So here you can see the surface, and you got the first colonists, and they're able to fuse their Glycocalyx together to form this exopolymer, and then they get channels of different kinds of, areas that are aerobic and some areas that are anaerobic, and different kinds of So here's an animation showing how biofilms form.
Clean, abiotic surfaces immersed in liquid tend to attract and concentrate nutrients, whereas biotic surfaces such as plant or animal tissue can themselves be a source of nutrients for microorganisms. A biofilm is formed when microorganisms attach to and grow on a variety of biotic and abiotic surfaces. The initial attachment is usually reversible and often involves attachment by flagella, fimbriae, or other cell surface structures. This is followed by growth of the microorganism and production of an exopolymer, which makes the attachment irreversible. Growth within the biofilm is not uniform. Channels develop which permit incorporation of fresh nutrients and oxygen. Metabolic activities of microorganisms in a biofilm can create changes in the microenvironment. For example, an oxygen gradient can develop that permits anaerobic metabolism near the interior and aerobic metabolism on the exterior. Nutrient and pH gradients are also produced. In the natural environment, biofilms often consist of different types of organisms that function together in the cycling of the elements. For example, breakdown of cellulose on the surface of a decaying plant may result in release of glucose and other carbohydrates, which supports the growth of non-cellulose degraders. Carbohydrates are converted to organic acids and methane by anaerobic bacteria, and the methane can then be degraded by aerobic microorganisms.
All right, so our concept check for this section: name the 3 parts of the flagellum. What is the difference between a run and a tumble? True or false, bacterial fimbriae are used for conjugation, and describe the differences between capsules and biofilms.